Hello, everyone. In this talk, I want to walk you through the idea of a confound continuum, which can serve as a two-dimensional confounder assessment for artificial intelligence in precision medicine. Brain behavioral predictive models can be useful for automated diagnosis, prognosis, or treatment choice, in other words, for individualized medicine. But it can also help to identify biomarkers or to enhance our understanding of brain function. In a basic neuroimaging workflow, machine learning algorithms are trained to learn the relationship between neuroimaging-derived features, structural or functional in nature, and a phenotypical or behavioral target such as age, sex, handicap strength, and many more. In the next step, the trained model will then predict the target from the features of a previously unseen test sample. To evaluate the model's success, often the predicted target is compared to the true target. For this talk, we will focus on handicap strength as a target. The reason for this choice is that handicap strength is an objective and highly reliable phenotype and therefore well suited for our exemplary workflow to rule out additional challenges coming along with other latent target measures such as intelligence or executive functioning scores. Additionally, handicap strength is an easy and cheap measurable assessment in clinical settings. Predicting handicap strength from cortical, subcortical, and cerebellar gray matter volume with a linear support vector regression and a decently sized sample from the UK Biobank, we reached a correlation between true and predicted target as high as 0.63. As a next step, one could tweak the features to see if other neuroimaging derivatives carry more signal about handicap strength. Using functional connectivity leads to a comparable predictive performance of 0.58. While for cortical thickness, the performance slightly drops to 0.36. Also, the choice of learning algorithm can be tweaked, both using a support vector regression with an RBF kernel or a ridge regression instead of the linear support vector regression results in similarly well-performing models. Tweaking the features or the learning algorithm are frequently used approaches in the field of neuroimaging predictive modeling. However, I so far left out a third very important component, confounders. For this presentation, we will follow a narrow ground definition of confounders, being variables that correlate with both brain-derived features and the phenotypical target, but are usually considered not relevant for the research purpose. What is often done to identify confounders is to statistically evaluate how much signal a set of potential confounding variables shares with the features of the target. We exemplarily correlated 130 potential confounding variables in the UK Biobank with both handicap strength and gray matter volume. In the handicap strength correlation plot shown here, we only depict the most important ones. If you're interested in all correlations, please come visit me at poster 816. The plot shows that mostly body composition measures, sex, and respiratory variables correlated with either the target handicap strength or the gray matter volume features. Measures such as length of the working week in the main job, systolic blood pressure, age, and bone density measures showed medium to small correlation with handicap strength or gray matter volume. Instead of statistical evaluations, often a standard set of confounders is used. Sex and age are thereby the most prevalently considered in the field. Mimicking this approach, we kept every step in the pipeline constant, but adjusted the features for sex and age in a cross-validation consistent manner. For the new model, true and predicted handicap strength only correlated with 0.08. This means that by just regressing out sex and age, our predictive performance drops from 0.63 to 0.08. In summary, together with the results from tweaking the features or learning algorithm, the drop in predictive performance when removing confounders means that confounders can have a higher impact on predictive performance than feature or algorithm choice. This change in predictive performance is not surprising considering the strong correlation of particularly sex with handicap strength. What remains unclear, however, is which of the two models is now the correct one. To dissect the dilemma a bit more, we want to distinguish between two different potential research endeavors. The first one we call high performance. In this heading, one uses few assumptions and all information and data to end up with a high performance. This approach is valid as long as the distribution of the training and the test sample match to guarantee proper out-of-sample predictions. The second one we call pure brain-behavioural link. In this setting, one is interested in specific relations between the brain and the target to gain actionable insights in the biological connection. This setup should also be robust to different settings as it clearly focuses on keeping samples independent and identically distributed. Nevertheless, we need to specify what purity conceptually means and if it ultimately can exist. To illustrate what I mean by purity, let's consider two exemplary confounding variables, length of the working week in the main job at the left and sex at the right. The set of biological attributes of length of working week is depicted by the rounded shape, and the set of biological attributes of sex is depicted by the triangle. The gray gridded area visualizes the biological attributes of the predictive problem space. In contrast to length of working week, sex quite overlaps in its biological attributes with the gray matter volume hand grip strength problem space, because sex influences testosterone levels, which in turn influence muscle growth, and the amount of muscles determines hand grip strength. In the pure link setup, with removing confounders, we aim for the raw or pure biology of the problem space. This pure biology is visualized by the red outline around the gray gridded area. We expect to keep all meaningful connections between gray matter volume and hand grip strength intact, but we want to get rid of influencing information of the confounding variables. Removing the non-overlapping variable length of working week, this works well. For the highly overlapping variable sex, on the other hand, we end up with the weirdly shaped red outline at the bottom right. The new set of biological attributes of the gray matter volume hand grip strength problem space is biologically ambiguous and not, or at least hard to interpret in its meaning. As a consequence, by removing confounders with a large overlap of biological attributes, we cannot reach purity, and this in turn means that we need to acknowledge a conceptual dimension of confound removal. Length of working week and sex have different biological attributes and hence a different overlap with the problem space. But there's of course more potential confounders for our exemplary predictive model. Using some of the example variables that we already saw in our statistical evaluations, we order them on an axis with increasing biological link or overlap of biological attributes with our gray matter volume hand grip strength predictive problem. Bone density measures, for example, most likely have different driving factors than both gray matter volume and hand grip strength, but the possibility of a biological link cannot entirely be ruled out. Going one step more in the direction of increasing biological link, systolic blood pressure does not have a clearly identified pathway, but potential common driving factors with hand grip strength. We can combine this conceptual axis, depicted in blue, with the statistical evaluations, depicted in red, to form a two-dimensional grid. It is important to highlight that the conceptual considerations are independent of the statistical evaluations. This becomes particularly obvious by the variables in the gray-shaded off-diagonals. With this two-dimensional assessment, we now have a very much better understanding of the different roles of potential confounding variables for our predictive problem. To decide which of the models is the proper one, we already know that the overarching research endeavor plays a role, but additionally, interpretation matters. With the vanilla model of no confound removal, we could claim that we decently predict hand grip strength. But, and this is important to notice, we cannot claim that we found neural encodings of hand grip strength. If data distributions match between training and test sample, such models can, for example, be useful for individualized medicine applications. With the sex and age adjusted model on the right, we could claim that sex and age encodings in the gray matter volume drive linear predictions of hand grip strength. Such models aim to enhance our understanding of brain function beyond overlapping other behavioral and phenotypical measures. The Confound Continuum aims to support informed decisions on confound removal in a problem-dependent way. It aims to complement a statistical perspective on confounders and highlight that biomedical and statistical validity are distinct concepts. It additionally wants to connect confound removal and model interpretation. In summary, we saw that confounders can have a bigger impact on a model than featured in algorithm choice. We also saw that there are two distinct possible research endeavors, high performance versus pure link. As a third point, we saw that in the pure link setup, there's a conceptual dimension of confound removal independent of statistical evaluations. In summary, it is important to think about statistical and conceptual implications in confound removal and draw respective interpretations. With that, I want to thank the CRC 1451 for funding this project and mostly all my supervisors. If you want to know more about the project, please visit me at poster 816. I want to thank you for your attention.